It's
That's right.
Postdoctoral research at MIT and Harvard Medical School, and also at the Max Planck Institute. After a postdoc, Hans left the academic world for a short period, 2 years, I think, working as a chief scientific officer Proteocure at Munich. He was recruited full professor at the University of Salzburg, approximately 20 years ago, I think, something like that. And during this period, Hans worked during a very long period on proteases involved mostly in hemostasis and coagulation, with brilliant mentors and colleagues from Max Planck Institute. I am thinking to Wolfram Bode and the Nobel Prize Robert Huber, but also, for example, at Milton Stubbs. Also, he performed fascinating, for me it's fascinating studies on the tricorn protease. He collaborated at this period with colleagues very well known by members of a proteostasis community. Michael Grohl, people from proteostasis know very well Michael. And also he had some interest with calicranes. He collaborated with friends such as Victor Magdalen, for example. His results were published in excellent journals like GBC, G-Mobile, Structure, PNAS, Nature, EMBO Journal, YACS, etc. Currently, he is a research group leader of a team working on diverse proteases. Basically, he used the approach he used was structural biology, protein chemistry, and enzymology, and his main topics are related to pro-protein convertases, bacterial collagenases, and obviously, it's subject today. He performed outstanding works on legumain with her colleague Elfriede Dahl. I think it's very important to mention Elfriede for this work. Hans is also, obviously, together with Claudia Brix, the main organizer of the famous Thier's Winter School dedicated to proteinases and their inhibitors, which was created more than 30 years ago by Hans Fritz and Vito Turk. And I have to mention, obviously, that the Winter School is now supported by Proteocure. And to conclude and make the transition with Hans talk, a short advertisement. I warmly recommend to everybody, very important, to read an excellent and still up-to-date review published 8 years ago in Biochimie. The title is Structure and Function of Legumain in Health and Disease by Dahl and Brandstetter. And you can find also an exquisite front cover of a corresponding directed issue. Please, Hans.
Thank you very much, Jill, for these very kind words, nice words. Thank you, Laetitia. Thank you all the team to invite me and give me the opportunity to share some of our work with you. Now I try to share also my screen again, and I hope it works as well as it did before. Let's see. Oops. How about that? Do you see the right screen?
It's okay.
It's okay. Cool. Super. So, yeah. So I wanted today to talk about the plan of CD cysteine proteases, where obviously also a very important member is the family of legumains, but there are others. So now my mouse, oh yes. So if we look into this clan of CD clan of proteases, and we look into the famous MEROPS database that we all appreciate a lot, we see important members or family members in this clan. And well, and some of them, all these families of course contain a number of members, and I highlighted here in green those which are strictly conserved in animals and mostly conserved in eukaryotes. You see that families of this clan are also found, of course, in bacteria. And I also should point out that as much as I cherish this MEROPS database, it's not complete. Because, for instance, this caspase, so this CRISPR-associated caspase family is not yet contained, but that's also important, certainly. I will focus now a little bit, or I will focus in my talk here on these more eukaryotically conserved members, legomine, caspase, and separase. Now, I hope these examples illustrate that these families are really important enzymes, and they decide literally about life and death of a cell. And as an example, I just mentioned separase. Many of you will know about separase. Separase is critical in triggering the anaphase on the cell cycle control, because upon cleaving the cohesin which holds the sister chromatids together, you can trigger this cell cycle step. So it's really important in that sense for birth of a new cell or two new cells in mitosis. So that's obviously very important and something that you really want to control, obviously. And also caspases are well known, maybe even better known. And they are also very critical for death, for cell death, for programmed cell death, apoptosis, just to name one. This is only one of many functions of different caspases. I think that is also clear, but it should make the point. It's really important functions. Now, caspases are a bit special because they are also special with respect to their substrate specificity, cleaving of the aspartic acid, which hardly any other protease will do. So that's makes it special. Actually, this is also the reason for its name. Cysteine protease is cleaving of the aspartic acid caspases. Right, so, and now there are, of course, different caspases. Caspases, again, is huge family. And most caspases are these cysteine proteases with specificity for aspartic acid, but some caspases are more equal. So there are these paracaspases, autocaspases, which have different specificities for arginine or lysine, for instance. Now, arginine and lysine is actually not, this is a typical tryptic specificity, so this is not so special. But they have the same fold. That's the same for them as the other caspases, and of course, high sequence similarity. Metacaspases are found and critical in plants, and they actually have similar functions in plants like caspases in animals. They induce programmed cell death. And then finally, of course, I want to briefly mention legomine and legomine's functions. Legomine has also many functions. But one which is probably most prominent is the one in antigen processing and antigen presentation. So obviously that's important for priming and maturing them T cells, and you know that. But for MHC2 presentation, so it's actually the left arm only that is important. And yeah, now where is this happening? Legumes are present mostly in lysosomes, endolysosomes, and there is this antigens are entering by endocytosis, and they are cleaved in these compartments. Now this is important because the pH in the endolysosomes is acidic, and it gets more and more acidic. So this is important also, and we will come back to this aspect later. Right. Legumins are also present in plants, and that's where the name originally is derived from, as one probably can guess. And it's also supposed to have a role in programmed cell death in plants. But this is not so clear, after all, one has to say. It's a bit controversial. Legumin, as I already indicated, of course, also shares the same protein fold, so it has a very similar structure. And if I say the similar structure, then I mean in particular this catalytic domain, which you see here in the center and here. And so that's the same fold, a 6-stranded beta sheet and such. So that's very similar. But of course, then there are extra domains which make differences. Also, I want to mention, of course, that legumin has a very prominent substrate specificity, and this is the specificity for asparagine. So it's cleaving after asparagine in P1, and that is the reason why it's also referred to as an asparaginyl endopeptidase, or AEP. This is a synonymous term for legumin. It's the same enzyme. It can accept aspartic acid at low pH simply because the aspartic acid gets protonated at low pH. So that's an overview of functions of these three families in the CD clan. Now I want to briefly look at the activation of these CD clan proteases, and that's interesting also. So starting with separase, there it is such that separase is actually produced and co-translationally folds with its inhibitor chaperone. This is the secorine, and you can see here, well, on the one-dimensional representation shown on the left, we see that the two proteins need to be co-translated, so you need them both, because the secorine, which is shown here in purple, actually binds to the separase and acts as a folding template, and of course also inhibits the catalytic domain, which is shown here in cyan. So this is then blocked, folded, and blocked, inactive. And how then do you get this separase active? Well, you need to make the active site accessible, and that is done by ubiquitination of secorine, and then cleavage and release of secorine that makes the separase accessible, the active site accessible to substrates. I should say this is not the whole story. This is one very important step, making the active site accessible, but it's not the whole story. You need additional factors to come in And this is so and so well understood, so it's only partly understood. Now, the second example are the caspases. How are they activated? The caspases need to form dimers, need to dimerize, and upon dimerization, you see here on the left, caspase 7 is a dimer, like this opposite direction, as you can see. So the L4 loops and L4 prime loops are from two molecules. And why is the dimerization important? Again, it's only upon the dimerization where these loops rearrange such to make the active sites accessible. So that's shown more in more detail on the right. So that's essentially what it does, how we understand it in the first place, making active sites accessible. And now this is, of course, one could think, so that's the story about activation, make it accessible. But it's not the whole story. And that is also very nicely indicated here by the metacaspase. The metacaspase, there is a propeptide bound to the active site, and it's perfectly bound like a substrate. But why is it not cleaved? And you can see at the electron density, it's intact. It's not cleaved. So there must be something, some magic bullet, which makes it active. And the magic bullet in the metacaspases, well, there are different types of metacaspases, but some of them is calcium. And you need quite a lot of calcium, in fact, millimolar concentrations of calcium. That then makes it active. Why is not so clear from the structure alone, because there is not even a structure bound calcium visible. So it's again indicating that the activation can be a bit more, is probably more tricky than we expect. Now, how about legomain? Legomain is activated by under pH control. So actually one can see that upon lowering the pH, the full-length protein is being processed, which means in this schematic that the C-terminal inhibitory domain is cleaved and then released, and that turns it active. So why the pH is important, that becomes clear when we look at the structure. So we first see that the zymogen, the full-length protein, has a C-terminal domain that binds onto the catalytic domain, and then that's a bit similar like saccharine in separase. So it binds and stabilizes actually also the catalytic domain, but of course it also prevents access of substrates to the active site. This must be inactive. Now, and how are they held together? They are held together by an electrostatic clamp. So there is negative charge on the catalytic domain, and I show that here with a surface, electrostatically colored surface representation, rotated for 90 degrees, in fact, here. And you see the negative charge in red and the positive charge of this inhibitory domain, L-sum domain in blue, the positive charge. That keeps them together, clamps them together. Now, if you now reduce the pH, you start to protonate the catalytic domain. The negative charge goes away, and such loosens the electrostatic clamp. So you can dissociate the two domains, and then things go on. So that makes it, that's how we understand why lowering pH can make it active. Again, this is only telling us something about making the active site accessible. That's the explanation so far. I also wanted to mention that this inhibitory domain, very similar as in separase and in other examples too, is actually also stabilizing the protein. So, and if we, in particular at neutral pH, the Elsem domain is important for the stability of the protein at neutral pH, because if we have the fully activated enzyme at a neutral pH, it's completely negatively charged. So an excess of negative charges, and that is kind of repulsive to each other, and that makes it very unstable. So whereas at lower pH, pH 5.5, for instance, the protein is perfectly stable. And we see that at this thermal shift assay, where we can measure the melting temperature, which is something like 65 degrees at acidic pH, but not even well defined at neutral pH. So, but the full length protein is of course stable at neutral pH too. So it's about this excess of charge, which is compensated by the Elsem domain. So if that is the case, now then we can also understand what is necessary to make this protein stable at neutral pH. Either you have it in this zymogen state with this Elsem domain on, that makes it of course stable, or you have another protein cofactor, for instance, that is also compensating and stabilizing these charges. So that's possible, and that's actually what we observe. And there is now this funny observation that this factor here, cystatin C, which is an inhibitor, is actually, so it of course inhibits the activity of the enzyme, but if you add the inhibitor and you go to high pH, say 6.5 or 7, then the cystatin will still bind to legumin and it will keep it stable. So if you now look at the activity or even reduce the pH where you can release the cystatin again, then you see that you regain the activity. But even at more neutral pH, what you find is that the enzyme is more active in the presence of the inhibitor than in the absence of the inhibitor, simply because in the presence of the inhibitor, the protein remains stable, whereas in the absence of it, it's unfolding. So that's a funny and nice observation, which nicely explains how important cofactors are for the protein to be active and stable at neutral pH. Only to test our understanding, we also modified these charges, say, by amidation of these carboxylate groups. So we would, ethanolamine, we would turn off the charges kind of, and then of course the protein gets perfectly stable. So it's really about this charge stabilization. That is the trick for that. So now we understand a little bit something about making the active sites accessible, and also the stability aspect that is particularly important also for legumin. But is this all? And do we fully understand the activity of these enzymes? And I would say no. And the reason for that is that we have here a special case. I wish to remind you of the canonical active site in a typical serine or cysteine protease, say catepsin, for instance. where you have a triad or a dyad of amino acids, the nucleophile, which is a serinoid cysteine, and neighbored in hydrogen bonding distance to a histidine, often at least, and oftentimes also a third residue like aspartic acid or asparagine. So this is important because the cysteine, as shown here in this example, needs to be deprotonated. You need to make the thiol a thiolate to make it nucleophilic, and only then it can attack the peptide bond, the carbonyl carbon of the peptide bond. That's important, and that's textbook knowledge. But it's not the case in these CD clan proteases. So this is an example legumin, but it's the same in caspases actually, where you have also your cysteine and your histidine. These are these two catalytic residues, but in between the two is the substrate bound. So cysteine and histidine are apart, like 6 angstrom apart to start with, but in addition, the substrate is in between. So there's no way how the histidine could assist that cysteine for deprotonation. So the mechanism must be different. That is not like in the canonical cysteine histidine dyad. It's different. So it's not this, but it's rather more this. The two residues are separated, and that, of course, then also brings up a few questions. First, to start with, how then is the proton abstracted from the cysteine dole, or must the cysteine be protonated to start with the reaction, or should it be rather be deprotonated? So that are questions that one could have. And I mean, just to again also validate our understanding, we actually mutated also here a neighboring residue neighboring to the catalytic cysteine. Here is a glutamic acid in human legumin and most legumins. A glutamic acid, and with a little bit of rationale, which is maybe not so important, the negative charge here essentially makes it such that the proton actually is more favorable on that cysteine. So it changes the pKa such that you find that cysteine more protonated. So we thought, how about fiddling around and changing this glutamic acid against a lysine, which is positively charged and should then have an effect on the pKa. And indeed, the K-cut is increased 4 to 5-fold. So it has a clear effect. So that tells us, yes, different things. Protonation is absolutely important. Second, legumin is not optimized to be a very fast protease. It could do better if that were the aim. So to better understand that, we also did some computational studies, density functional calculations, and where we try to understand what exactly is going on. And so now, and you can there model how protons are moving. And what we find as the only consistent solution is that we actually need the cysteine to be protonated to start, but then the proton abstraction is done by the P1' amide, and at the same time, synchronously, the attack must take place of the thiolate on the carbonyl peptide. So we see that at the same time. So that's different from the classical canonical proteases. this is done not step by step, but synchronously. So that's how it is working. And it's absolutely critical that you have a proton on the cysteine. To show you that, I have here a video. You see here that cysteine, which is protonated to start with. You see the peptide bond, the circle to green encircled atoms, or the carbon and the nitrogen, and now it will attack the carbon, and the proton will jump over to the nitrogen. So you see it. So it's a synchronous movement. The cysteine approaches the peptide bond, and in a synchronous action, proton is accepted by the amide, and the carbonyl is attacked by the S-. So that, of course, leads to bond rupture. That's how we then understand that. And of course, then we are still have the non-primed product bound to the enzyme, so the thioester bond, and of course that needs to be released too, and that is, as these calculations show us, done by the catalytic water, of course, and that's the classical way. But what is interesting is again, there is a difference. So how, namely, we end up, the histidine, the role of the histidine. The histidine is, well, maybe taking the proton ends up on the diol. We have a geminal diol, so it's not sitting at the histidine to start with, but it's only then later on. So the histidine has a role in positioning the catalytic water. So what we see in a summary now, how the proteolysis action works, is it's substrate assisted. The substrate is abstracting the proton, and that's how the reaction can take place. So we can answer the two questions. The P1 amide is abstracting it, and the cysteine needs to be protonated. Now, a further aspect I want to discuss is the reverse reaction. So there are actually, we find also a reverse proteolysis reaction. So, and we found that in this complex with an inhibitor, and what we found there was an interesting observation that cleavage occurred in a pH-dependent way. So we had this complex, and we found cleavage, complete cleavage at low pH, but not so at more higher pH of pH 5.5. So that was surprising, because for fluorogenic substrates, AMC substrates, with asparagine P1, you would expect a maximum activity at pH 5.5, but that was not the case in an ascent peptide in this complex. So we have seen more complete cleavage at pH 4, not 5.5. That was surprising. So looking at this more systematically revealed then quickly that actually you cleave at pH 4, and then you take this sample and increase stepwise the pH, and you find resynthesis apparently. So that's kind of funny. And actually, to further prove that, we also then introduced here a modification with MMTS, a thiomethane group, would block the catalytic cysteine. And now we see actually ligation independent from pH. So we see that at all pHs. So that's kind of funny. So that means apparently we do have ligase activity over all pH range, but it's of course the protease activity is dominant at lower pH. But if we block the protease activity by blocking the cysteine, for instance, by mutation or by chemical modification, then we see only ligase activity. So now let's look at an example in plants where this actually plays an important role, because there are certain peptides which are actually released from larger proteins, from plant albumins, and there you can release such a cyclotide, and you cleave that out, and what you observe is that the cleaved fragment then can cyclize. So that's cyclotides, and these cyclotides, for instance, have then functions as inhibitors and other functions. So of course, the plant legumes are catalyzing this reaction because we have an ASN or aspartic acid at one site. So that's what legumes can do. Now, we were looking at how would that work, and we also then tried to look at these computational studies. So how can that work? Well, if we have a cleavage, then we have always the thioester intermediate. And usually we would then have a catalytic water which would release the bound product, non-primed product. But if we can make sure that the catalytic water has not so well access to this site, then a new amino terminus can act as the nucleophile and release the product. So we would have aminolysis rather than hydrolysis, and that, of course, would do nothing but a synthesis of two peptides. Transpeptidation, it's also how it is called. So, and that's favored by legumain, and to understand that better, we will see it's important. The enzyme needs to prevent the catalytic water to hydrolyze it, and that's done by keeping the primed product or primed substrate, the primed product rather long on its side to block the access. We will see that in a second. So here is the active site, and we can look at different legumains, and different legumains have a different strong ligase activity. If we look at strong ligases, they have very pronounced binding sites on the primed site for the cleaved products, where they will still bind and prevent the catalytic water, the access of the catalytic water. So that's how it kind of deaccelerates the access and gives the transpeptidation better chances to occur. So that's and we also tested that then, of course, for instance, by using this such a peptide which has the ability to cyclize in principle, and then we tested the primed product that is then cleaved after the aspartic acid in that case, and we tested here the different amino acids. And what we find is that the P2 prime position, which is here this leucine, is very important. You need a hydrophobic residue to have it stick at this site. So we tested that, and if we after cleaving here, after this aspartic acid, we can, for one, get the cyclic form, where the glycine, the N-terminal glycine then is doing this aminolysis here. Or of course, we can also have a linear product. That's the classical hydrolysis. So that can happen too. And when we look at this by and analyze that by mass spectrometry, then we find that it's and varying these peptides, amino acids, and we find that leucine is very important. It's the best residue here to optimize the cyclic product. Right. So so that's also here shown in a small summary that the product, the cleaved product, if it's released fast, then of course access is easily for the catalytic water is easily possible, and you will find hydrolysis. If the primed product remains there, has a long residence time, then access for the catalytic water is not so easy, and you have chances to form this cyclization, the aminolysis, where the N-terminus then releases the substrate from the active site. That's indicated here. So we have the N-terminus, which then has the chance to attack the thioester bond and then release it. We'll break the thioester bond and let the ligand release from the enzyme. Yeah. So we find that leucine in the P2 prime position is very important to increase the residence time. Now that's of course also relevant for a number of examples. One is for actually the activation and inactivation of legumin itself at neutral pH. We find that a already activated legumin can go back to the zymogen prolegumin state, which is consistent with this pH dependence. Also, I wish to revisit now this somewhat strange or enigmatic activation that we have seen for metacaspases and their effect with calcium. So, what is important to note is that this linker, this green linker, is bound to the active site, both on the non-primed and the primed site. So these are prerequisites that ligation of cleaved substrates is possible. So if you cleave here, you can in principle religate here. So that's possible. So that's one aspect to consider. And another aspect, why then now with calcium, in the presence of calcium, we can see that cleavage is a different aspect. So there must be, we suspect it must be relevant to this pH effect or the pKa effect that we have seen. And we actually then also tested that a little bit, and we look at different legumin variants for which the ligase or protease activity is well recorded. Then we see that this nicely fits with our model. For instance, there is this legumin variant from France, butylase 1. Butylase 1 has exclusively reported a ligase activity. Its pKa is, when we calculate that, is low at 4.6. Butylase 2 is a very closely related isoform from the same plant, but it's interestingly has only protease activity, no ligase activity reported. We do the calculation of the pKa although it's very similar by structure, of course, the pKa is much higher, 7.6. Now see, if you just compare those two, 7.6, so you find this cysteine, the catalytic cysteine, protonated at, say, around neutral pH, whereas for butylase 1, with this low pKa, you find the cysteine being deprotonated for pHs higher than, say, 5. And that's why you cannot observe any protease activity. That's exactly what our calculations also showed. So, and we can go through this list, and it is consistent in this list, and there are interesting examples where the activation mechanism is unclear. And I would propose that it is this pKa dependence that can be tuned by the environment very much, and that turns these enzymes active. And also, of course, then that also opens the question of where do we have rich activity, because at different pHs we can have protease or ligase activity. So to sum up at that point, there is this unconventional, non-canonical catalytic diet, and it makes life as a protease harder, and it certainly serves as a safety net, an extra safety control. And we see in the examples that I showed you that activation in the CD proteases, of course, requires at least two steps. One is making the active site accessible. That was cleaving the secorine in separase, or opening the loops in caspases, or cleaving and releasing the C-terminal domain in legumain. But in addition, there is this aspect of the pKa tuning. You need to have the pKa such that you can really have a protonated cysteine. Otherwise, you will not find any protease activity. You would find ligase activity, most likely. But ligase activity not only requires this pKa match, but also, of course, requires a primed substrate to bind, and that they find each other. So not every substrate will be able for ligation. That is very clear. So, but you also need to exclude the catalytic water from the active site. Yeah. So that was what I wanted to show you today, a little bit about this weird, and I would say a little bit overlooked activation mechanism and strange catalytic diet present in the CD clan of cysteine proteases. I wish to very much thank people in the lab, Elfriede, Hasneem, Maya, Florian. They all have done extremely helped a lot, and of course cooperation partners. And I wish to thank you for